Hello, and welcome back to another lecture video. So, my name is Carlos Brian Francia, and today we will be talking about the nervous system. We will be looking at the central nervous system and the peripheral nervous system. The central nervous system, that composes of the brain and also the spinal cord right here. Branching out of the spinal cord is the peripheral nervous system. So here we have the little branches going this way, branches going that way, etc. Within the central nervous system and the peripheral nervous system are two types of cells that are very, very important. The first one are going to be called neurons, and the second one are called glial cells. Glial cells. We'll talk about glial cells later, but for right now I want to focus on the anatomy of the neurons and the differences of how they're called in the central nervous system and the peripheral nervous system. Here we have a neuron. So a neuron is present within the central nervous system and also the peripheral nervous system. Within the neuron, we have the cell body. That is the body of the neuron. Scientists are really good at naming things. Extending out of the neuron are called the processes or the processes, as however you like to pronounce it. But before we talk about those processes, I personally find it easier when I imagine these little thingies coming out of the neurons to be trees. So here on the ground is going to be the cell body. The trunk right here, that's called a process. And that is similar to this little tree trunk right there, to this guy. Extending out of the process are the branches. They are the little branches of the tree. And likewise, extending out of the process here are the little branches. And they are called dendrites. In fact, dendrites come from the Greek word branch, because they also thought that these little dendrites kind of look like branches from the tree when they were discovering the anatomy of the neuron. If we zoom in even closer, and let's do that in a different color, if we zoom in even closer on the branches, we notice these little items right here, and these are called synapses. So remember, when you're studying the muscles, the synapse will propagate the action potential into the myofibrils, specifically the sarcomeres, and help the muscle generate a contraction. Well, the branches on a tree, they have leaves, and these leaves are, well, they outnumber the branches. And so the synapses will always outnumber the amount of dendrites in a neuron. So here in this neuron, we can say that there are how many processes? 1, 2, 3, 4, 5, 6, and then 7. But there's something different about this large process. The largest process, or I guess you can say the largest tree in the park being the neuron, that is going to be called the axon. The neurons will always have one axon that is larger, or I guess will always have a process that is larger than the other processes. Now notice that we kind of go from a fat section, this section right there, into a thin section. When we go from a fat section to a thin section, this section itself is called the hillock, specifically the axon hillock. So this is the axon, let's write it over here, hillock, and that's a K. And that's from this section to that section. We can also notice that there's something weird right here, and that's called an oligodendrocyte. And this is one of the glial cells. We'll talk about glial cells in a little bit, but there are more glial cells, and they have different names and different purposes. For right now, the oligodendrocyte acts as the insulator for the axon. So you can think of it being like a parent. So the parent doesn't want the little child to go outside and play without a jacket. I mean, it's like 20 degrees outside. You want the baby to have a jacket, right? And a hat. And so the oligodendrocyte makes sure that the axon has its little jacket. The jacket being the myelin sheath. So this is going to be the jacket. And what the jacket does, what the myelin sheath does, is that it insulates the action potential, or the electronic impulse, from the neuron, and it basically allows the action potential to go to the next target. So let's say that here we have a muscle. So let's say that this is in the peripheral nervous system. We have a muscle, and we're going to draw that in red. So let's just say that's a little square is a muscle. The action potential will be carried from the neuron, and it cannot escape the myelin sheath, and so it's going to jump, and it jumps over here, and it jumps over there, and it goes here, and it jumps here, and then it can propagate into the muscle cell in this example. The gaps between the myelin sheaths are called nodes of Ranvier, and that allows this jumping action that you see here. This is a jumping action that allows the nerve impulse to travel from point A to point B in a fraction of a second. And it also conserves the amount of voltage that we have. So let's say we have 30 millivolts right here. We're going to have 30 millivolts right there, or a value very close to 30 millivolts. Hypothetically, if we did not have the myelin sheaths, then we would start at 30 millivolts, and then we would have, let's say, 26 millivolts. And instead of taking a fraction of a second, it would take maybe a quarter of a second or half a second instead of 0.00001 seconds. So it speeds up the action potential, and it also preserves the amount of voltage that we have. The jacket that is given by the oligodendrocyte, the myelin sheath, that's actually a bundle of lipids. So this bundle of lipids wraps and wraps and wraps and wraps and wraps around the axon. When we look at a 3D model, you will notice that the myelin sheath is just a very, very, very large piece of lipid that is finely wrapped multiple times around the axon to make a very compressed and dense jacket of fat. And dendrites are the way that the neurons communicate to each other. So here, let's say hypothetically, instead of this muscle cell, it was a neuron. And so an action potential can go here and travel from this neuron. Well, that's wrong, the way I'm drawing it. So it's going to jump, of course. And instead of going to this muscle cell, it goes to another neuron, and it repeats. Within the central nervous system, we have gray matter, and we have something called white matter. If we take the human brain and we cut a cube out of the human brain, we will notice that there's a tan color found in this section, in this example. So it's this little tan section, and that tan section is gray matter. What gray matter is, it's neurons that have a lot of dendrites. So in this picture right there, this neuron has dendrites, But let's say it has even more dendrites. So let's say it's right here, there, more and more and more. And you know what? Let's add some more processes. So we're going to add this process. We're going to have more dendrites. So let's say we have a lot of processes within the neuron. The reason why we want to have a lot of processes and subsequently a lot of dendrites is because the more dendrites we have, the more neurons we can communicate with. So instead of only communicating with 3 neurons, this neuron can communicate with 16 neurons at a time, maybe even 100 neurons at a time. The reason why we want to have a massive connection between neurons is because we want to have a reliable way of relaying information from the brain. So if the brain senses something, we want that information to quickly go from point A to point B or to point C. The more dendrites we have, the faster that connection will be. So a neuron at point A can quickly communicate to point B, and that can quickly communicate to point C, and that can quickly communicate to point D, etc. So the more dendrites you have, the more connections to other neurons you can do. And that will take a fraction of a millisecond to go from point A to point B, etc. Within the white matter portion of the brain, we also have neurons. However, these neurons don't have that many dendrites compared to the gray matter. However, the axons within the neurons within the white matter have more myelin sheaths compared to the neurons in the gray matter. So notice both in the gray matter and the white matter, the neurons have axons. All neurons have axons. However, in the gray matter, the neurons do not have that many myelin sheaths. So let's say there's only 2 myelin sheaths, hypothetically. In the white matter, you would have like 16 or 20 myelin sheaths. Because the myelin sheaths are made of lipids, the whiteness of the fat in the myelin sheath creates this whiteness that you see in the white matter. So let's say that you're cooking chicken, you have the raw chicken, you notice that there are globs of fat on the skin, and that little glob of fat is white. Because you have such a high concentration of myelin sheaths, which are made of lipids, that myelin sheath show the whiteness in the white matter. On the right, we have a cross-section of the spinal cord. I don't want you concerning yourself about the names right now. For now, I just want you to look at the structure. Within the spinal cord, we have the opposite orientation of gray matter and white matter. In the brain, the gray matter is going to be on the outside, and the white matter is going to be on the inside. In the spinal cord, the white matter is on the outside, and the gray matter is on the inside. The reason why we have opposite orientation is because the white matter, since it has so many myelin sheaths on the axons, it can effectively transmit information from the spinal cord, or I should say from the brain, into the peripheral nervous system. So running out of the spinal cord are little nerves. And these nerves are part of the peripheral nervous system. So the brain can tell you to move your left arm, and that's going to go from the brain. So it goes from the brain, is going to travel into the white matter of the spinal cord, and since the spinal cord, or I should say, since the white matter has so many myelin sheaths on the axons, it can relay this information into the nerves that go into the muscles in your arms and permits movement. If we touch something hot, like on a stove, well, that information goes from the fingertips into the nerves from the peripheral nervous system. It goes into the spinal cord, and now that information can go into the gray matter. So the gray matter are neurons with a high concentration of dendrites. The more dendrites you have, the more neurons you can communicate with, and therefore the information of hot or cold can effectively be transmitted to other neurons and travel really, really quickly into the spinal cord and eventually the brain. So that's why we have a opposite orientation of white matter and gray matter within the spine as compared to the brain, in which the gray matter is on the outside and the white matter is on the inside. In the nervous system, there can be different names between the same collection of items. For instance, in the central nervous system, the collection of neurons, let's say that this is a neuron and this is also another neuron, and another one here, and another one here, and another one right there. This collection is called a nucleus. It's called a nucleus, and that's when you're calling it in the central nervous system. So if you look at the brain and you see a collection of neurons, that collection of neurons is called a nucleus, since it is found within the central nervous system. However, if we were to take another collection of neurons, let's say that these 4 circles are neurons and they're found in the peripheral nervous system, this same collection will be called a ganglion. Now let's erase the board. When we have a bundle of axons, so let's say we have another axon here, let's do it in Mint. We have another axon here from a different neuron, another axon here from a different neuron, another axon here from a different neuron. Let's just do the neurons right there. This whole bundle of neurons will be called a tract, and that's in the central nervous system. However, if you're in the peripheral nervous system, and you have some neurons here and there and there, and for some reason they all accumulate in this section here, well, this whole bundle of neurons are called nerves. So within the peripheral nervous system, the bundle of axons are called nerves, and the bundle of axons are called tracts in the central nervous system. And that allows scientists and medical physicians or whomever to effectively name the collection of neurons, whether that be in the central nervous system or in the peripheral nervous system. A prime example of the difference in names between the central nervous system and the peripheral nervous system can be found within the optic chiasma. So the optic chiasma will act as the border between the peripheral nervous system, this being the eyes, into the central nervous system, this being the thalamus. So the thalamus takes in inputs from the outside world. It kind of tells the brain what to do with it. So let's say that you see something. Let's say that you see Zendaya out in public, and only Zendaya. I don't know who that is. It's not like I care. So this image stimulates the nerves in the eye. So notice that the nerve is going to be a collection of axons of the neurons. But since we're in the peripheral nervous system, the collection of axons of the neurons are called the nerve. So now the nerves will send an action potential down here. When it crosses the optic chiasma, we become the tract. So now the word nerves become the word tract. So we still have a collection of axons, but since we converted from the peripheral nervous system into the central nervous system, that collection of axons is now called a tract. Also, since I remember studying this in the MCAT, images or stimuli from the left eye will cross over within the optic chiasma and be processed within the right side of the brain. And subsequently, an image seen within the right eye will cross over within the optic chiasma and be processed within the left side of the brain. And the MCAT never leaves you. It never leaves you. Sometimes I still have nightmares. Going back to the big picture, we have different types of nervous system. we have the autonomic nervous system. So we have the autonomic nervous system, and we also have the somatic nervous system. The autonomic nervous system is the part of the response to a stimuli that you don't control. So let's say that you ate a taco. So the taco goes here, and it goes into the organs, which is not pictured, and then the body will have a collection of nerves that innervate the organs and will contract and decontract the stomach and the small intestines to move the food and to digest the food, etc. You don't purposely control that. The brain controls it. So the central nervous system can do autonomic nervous system. And really the peripheral nervous system can do autonomic and somatic, and so can the central nervous system. When we're looking at the somatic nervous system, this is something that you can control. So let's say you want to move this thumb this way or that way. Well, the brain sends information along the spinal cord, and then it goes into the nerves that go into your hand, and now your thumb can move in this direction or that direction. And so the somatic nervous system is something that you can control, The autonomic nervous system is something that you cannot consciously be controlled. Going back into the neurons, we have 3 different types of neurons. First is a unipolar neuron, and this is not really present within humans. It's actually present within the invertebrate, kind of like a fly or starfish. So we're mainly just going to ignore it. I just included it because it's there. It's like, whatever. Next, we have the bipolar neurons. So this is the bipolar neuron. Notice that here we have 2 processes. That's why it's called bipolar. Bi means 2. Bi. So we have 2 processes, and we have these dendrites on the opposite ends. Now, between the processes, one of them will be larger than the other. So let's say, for this example, just randomly, I'm going to assign the right one as being the larger process. And so therefore, this would be the axon, while the other one would be the process and the dendrites. So this one would receive information, and this one would relay the information to either a different neuron or to a cell. This is really present within the olfactory system, and also part of the retina system. So let's write this out, the retina system. So you can think of it as part of the lower brain where the nose connects to the brain. Not the nose connecting to the brain, but the nerves from the nose connecting into the brain. That would be part of the olfactory system. And also the eyes. So the retina system would be using the bipolar neurons. And then the rest of the neurons in the body are going to be multipolar. So we have different processes. We have multiple processes and multiple dendrites and only one axon. So this axon is larger. It's a larger process than the other ones, and it's going to have your little synapses at the end. So that's pretty nice. But you know what is even nicer? Seeing things in 3D. So now we're going to increase the budget of this production video, which is $0 because I don't have a budget and I don't make money. So now we're going to see the glial cells. We're going to be introducing you to the glial cells. And this is going to be really cool. Here we have the nerve tissue. Now I know that it looks like a very busy image, so let's kind of hide different items. First, we have the astrocytes. So the astrocytes are given the name because they look like little stars. Astro meaning star. Now notice if we zoom in on an astrocyte, this astrocyte has many processes. This little tree trunk looking things are processes. Astrocytes, they work well within the nerve tissue and do a multitude of things for us. However, for this purpose of the video, I'm going to limit what we know. But what we should know is that they help regulate the transmission of electrical impulses. So recall that an action potential relies on sodium and potassium. Well, when you have an action potential, the potassium goes out of the cell. Well, we need that potassium inside the cell, or we need that potassium to disperse. And what allows, or what facilitates this, is the astrocyte. The astrocyte will speed up the process in which potassium is either removed from the area or goes back into the cell. So let's say that we have the neuron. So let's say that my cursor is a neuron. The neuron will release potassium because of the action potential, which we will later learn about in this video. The ions of potassium will go into the astrocyte because the astrocyte is highly permissible to potassium. The potassium will then be processed within the astrocyte. This is great because we don't want to have a high concentration of potassium in the fluid surrounding the astrocytes. If the fluid surrounding the nervous tissue has a high concentration of potassium, that could lead to epileptic seizures, which is the overstimulation of the nerves inside the brain. And so astrocytes are really nice because they help maintain the amount of active potentials occurring at once. In addition to this, astrocytes can do gluconeogenesis, which means the creation of glucose, and that helps feed the nervous tissue. They can also provide lactate to the nervous tissue. Going back to the main structure, let's look at this right here. Let's look at the oligodendrocyte. Oligo means few, and dendrocyte means branching. Well, dendro means branching, and cyte means cell. So this is few branching cell. Let's hide some certain items. So let's select this, and we're going to select the axon, and might as well select the little nerves, the little glial cells. So here we have the oligodendrocyte, and notice that it's going to be branching into different axons. So again, think of a mother putting a jacket around her child. She may have 1 or 2 children. She may have 3. Basically, what the oligodendrocyte does is that it will facilitate the insulation of the axon. So it will create this little jacket of myelin sheath around the axon. Do you remember when we were talking about the muscle cell or the muscle tissue and we had the Schwann cells? Well, it's very similar. For the oligodendrocytes, the Schwann cell does something similar. Within the central nervous system, the oligodendrocyte creates the myelin sheath for the axons. Within the peripheral nervous system, the Schwann cell creates the myelin sheath. While we're at it, let's look at this myelin sheath. The myelin sheath is made from lipids, which gives it its nice white color. This lipid will go around and around and around the axon multiple times, and it's very, very compact. So when the electricity goes from one end, let's say it's right here, to this end, it's because it cannot go through this myelin sheath. It's easier to go from here to here than it is to go from here all the way through there. And these little gaps, again, of course, are called nœuds of Ranvier. And so to recap, oligodendrocytes create these little myelin sheaths around the axons. And Schwann cells do the same thing, but they are found in the peripheral nervous system. Going back to astrocytes for a quick second, astrocytes basically maintain the area in which the other glial cells operate. So they make sure that the potassium concentration is leveled, they make sure that everybody's being fed with glucose, and that's in the central nervous system. Within the peripheral nervous system, instead of having astrocytes, you will have satellite cells, and satellite cells monitor the area and make sure that everything is running smoothly. Now, the following two glial cells do not have any other cells that are similar to it in the peripheral nervous system. So I cannot say, oh, this cell acts similarly to this other cell in the peripheral nervous system. No, it's not like that. So the next one that we're going to be talking about is called the microglial cell. So let's hide the others. And here, the microglial cells will do what? They will form, or they will act like macrophages. So macrophages are special types of white blood cells that will eat bacteria and other pathogens inside the blood. The microglial cells will do that for the central nervous system. So let's say that you have a traumatic head injury. The microglial cells will connect to each other. They'll really go to the injury and start creating a concentration of microglial cells. And that is in response to any incoming pathogens or bacteria that may enter the central nervous system. And we never want that to happen, but sometimes it does happen. And so we need specialized cells to eat anything that's bad. And we need specialized cells to clean destroyed cells. So they kind of function as both the cleanup crew and as the warriors for the central nervous system. The last glial cell that we want to talk about is called the ependymal cell. So it is the ependymal cell. On one side of the ependymal cell are cilia. So these cilia help move the cerebral spinal fluid. The purpose of the ependymal cell is to create cerebral spinal fluid from blood. And the way that we do that, or the reason why we do that, is because we have something called the blood-brain barrier. We don't want the brain to have access to blood that may be contaminated. So within the blood-brain barrier, blood is converted into cerebral spinal fluid. Glucose, which travels within the blood, can easily pass through the blood-brain barrier. Oxygen can also pass through the blood-brain barrier, and so can other gases. And so when pharmaceutical companies are creating drugs that act on the nervous system or the brain, they have to have a mechanism in which the drug can pass easily through the blood-brain barrier. Also, the cerebral spinal fluid that the ependymal cells create provide nutrients to the brain, and they also form kind of like a cushion for the brain because parts of the brain are hollow. So now let's pull up a picture of the brain. Here we have the brain in two different pictures because I want you to have different ways of viewing the brain. On the very far left, we have the capillary blood. So specifically, the cerebral spinal fluid is created within the choroid plexus. So that is called the choroid plexus. And this is that region right there. So in this region, the capillary blood goes into the brain. Well, not the capillary blood, but the capillaries are in the brain. And we don't want just pure blood going into the brain. We need to filter it out. We need to filter it for the blood-brain barrier. If I have some bacteria here, and hypothetically we didn't filter out the blood, the bacteria would infiltrate the brain and cause havoc. That's actually cause of the meningitis. So the meninges of the brain become infected with bacteria, and it causes inflammation because there is a failure within the blood-brain barrier. So instead, the capillaries will meet with the ependymal layer, and small bits of blood, let's say that some of them leak, quote unquote, leak into the ependymal layer, and the ependymal layer converts it into cerebral spinal fluid. So we have the conversion of capillary blood into the cerebral spinal fluid for the blood-brain barrier. But why do we need cerebral spinal fluid anyways? Well, when the brain is forming, it has these little areas that are hollow, and these areas that are hollow are called ventricles. And right now, we don't have to concern ourselves about the names of the ventricles. I just want you to know that the brain has these little hollow parts called ventricles. Well, we need to fill those hollow parts with fluid, and so the cerebral spinal fluid will accumulate within the hollow parts of the brain. And again, the cerebral spinal fluid will have oxygen, it will have gases, it'll have nutrients, it'll have glucose, but it will not be blood. We don't want any pathogens found in the blood to make its way into the brain. We want to filter it out and convert that blood into cerebral spinal fluid. And not all of the capillary blood will become cerebral spinal fluid. I'm going to make up some numbers. Let's say that 99% of the capillary blood within the choroid plexus remains capillary blood. The cerebral spinal fluid is only made up of 1% of that capillary blood. I don't know the exact numbers, but I'm just saying that not all of the blood within the choroid plexus capillary becomes cerebral spinal fluid. It just kind of leaks out into the ependymal layer and converts into cerebral spinal fluid. And on the far right picture, we have the areas that are hollow. We have this area that's hollow, and some of this right here, and this part right here, etc. So that just shows you how hollow the brain can be. And those hollow points are called ventricles. And the brain has those hollow portions when the embryo is forming. Now let's take everything that we learned so far and put it into one picture. In this example, let's say that this person is getting ready for medical school, like me. And personally, when I start my mornings, I take a shower. So the way that I take a shower is that I turn on the water and I let it warm up. And now I'm going to check if the water is warm enough. So here we have the stimuli, or you can say stimulus. Specifically, is it hot, or is the water, or is the water cold? Let's say that for our instance, hot, the sensation of hot on the skin will be about 100 degrees Fahrenheit. And cold would be about 60 degrees Fahrenheit. So, of course, you've touched a warm or a hot cup of coffee. It feels hot on your skin. And you've touched ice cubes, which feel cool on your skin. And that is due to a threshold. So we have a threshold. So that sensation is due because it reached a threshold. Maybe it has to reach a threshold of 100 degrees Fahrenheit on the hand to register as hot, or it has to go below a threshold of 60 degrees Fahrenheit for it to register as something being cold. And that's a very oversimplified example or oversimplified explanation for this example. But let's say that we hit the threshold for hot. So now we generate an action potential from the sensory nerve. So the sensory nerve senses the stimulus, the threshold is broken, and now we can propagate an action potential, which starts right here. So this action potential will jump across the nerves. So notice that I said nerves. Nerves are the collection of axons of the neurons within the peripheral nervous system. So this action potential goes along the nerves into the spinal cord. So first it goes into the white matter of the spinal cord and the gray matter in the spinal cord. This is the opposite in the brain, in which the gray matter is on the outside and the white matter is on the inside. The reason why it's called white matter is because it has a high concentration of myelin sheaths, which are made from lipids, and lipids are white colored. Now it goes, this action potential goes, and it connects into the thalamus. The role of the thalamus is to take in sensory inputs and to relay them into the cerebral cortex. So the cerebral cortex is right here on the right portion of the brain, or on the left portion as well. It's really just this upper region. But the purpose of the cerebral cortex, so we're just going to call this cerebral cortex, is to have opinions about the stimuli or the information. So I have my hand under the water, it's hot, and I said, yes, this water is hot, but do I really want to take a shower? Do I want to take a shower in this kind of warm water, or do I want to increase the temperature of the water? Or matter of fact, do I even want to take a shower? Can I go eat breakfast or watch TV or just stay in bed all day? So the cerebral cortex has opinions about the stimuli of the world. Let's say that the water is not hot enough, so I want to move my hand to increase the heat of the water by turning the knob. The cerebral cortex says, you know what, the stimuli is not enough, let's make the water hotter. I'm going to move my hand. So the cerebral cortex sends information, and this information goes down the what? What's the difference between nerves and what do we have? We have the collection of axons in the brain called the tract. So now the information goes from the cerebral cortex into the tract. So now we're going to use a different color. It's going to propagate around here, and it executes a motor command, moving my hand to turn the knob to increase the water temperature, and it's going to go down into the spinal cord, from the white matter into the gray matter, and from the gray matter into the white matter, and it's going to go down to the what? To the nerves, because we switch from the central nervous system to the peripheral nervous system. And now it tells the muscles in my hand to move, and specifically to turn the knob this way to increase the temperature of the water. And that is the propagation of information of the nervous system. But notice that I did say action potentials. So now I have to tell you and teach you about action potentials. And you know who's an expert on that? Well, not really an expert, but me from 2021 when I was studying cell biology. So we will have past me teaching you now about action potentials and how it propagates and how it's formed. So enjoy. And now we begin the portion of the lecture that focuses on membrane potentials. So membrane potentials, they are governed by a permeability of specific ions. So a membrane potential. And from now on, I'm going to be calling membrane potential MP. Okay? So the MP of a cell is really dictated by how many ions are in the extracellular side and the intracellular side. And the distribution of ions on either side, it will actually create a potential. So let's say that we have too many sodiums outside of the cell and a little bit of sodiums inside the cell. Now, because there is a difference in charge, there are more charges outside than there are inside, we can actually create a potential. So we can have a greater positive charge right here and a small positive charge there. Now, the system will try to equilibrate itself. It will try to go into equilibrium. And when this happens, we can have a potential. And so what you see here are these little curves and these little strips. And this curve is called an action potential. So this is called an action potential. And this action potential is the entire basis of movement. So whenever your muscles are exercising, or whenever you're walking, or when you're breathing, you have trillions of action potentials going on in your system, and that allows you to move. And an application of an action potential is actually the Venus flytrap. So this is a Venus flytrap, and you may know that a Venus flytrap is a plant, but you're probably wondering, hey, I thought plants couldn't move. Why is this Venus flytrap able to eat a fly? Well, most likely it's hungry. I mean, I would bet that it's hungry, that's why it's able to eat a fly. But actually, it uses action potentials to capture its prey. So whenever a fly touches a little hair right here, the action potential is triggered, and so we see an influx of sodium entering the cell. And when this happens, we actually depolarize the membrane. We'll talk about that later. And when the membrane is depolarized, it allows the fly to be captured by the Venus flytrap. And so now we will watch a video of the Venus flytrap in action. And this was created by the BBC, the British Broadcast Company.
The Venus flytrap. Like the sundew, it makes itself very attractive, oozing nectar across the brim of each leaf. But any visiting insect had better watch out for these 6 tiny hairs. This fly has to tread carefully. If it strikes one hair, it can carry on feeding, but a timer has been set. A second strike in less than 20 seconds, and the fly is doomed. An electrical impulse is triggered and the leaf snaps shut in just a fraction of a second. The tips lock together like prison bars. If the fly is very big or very small, it may just manage to escape. But most are trapped and die. Ten days later, the trap reopens. All that remains is a husk. The plant has finished its meal and resets itself for its next victim.
I hope that fly is okay. I mean, he looked like he was having a bad time. He was struggling. You know, if that were me, I would have escaped. I'm just built different. So this is an action potential. And here we have a mixture of sodium and potassium. And so this dance that sodium does with potassium will create an action potential. But before we can get into what an action potential is, I have to introduce an equation. Now you don't need to memorize this equation, it's just there to help you understand what's going on and why we have a negative number. So over here we have that the membrane potential is going to be equal to 61 millivolts multiplied by the log of, and let's just focus on one ion. Let's say it's the log of the potassium inside the cell. Let's say outside. Outside of the cell, divided by the concentration of the potassium ions inside the cell. Now within a cell, there is more potassium inside. So this number, let's say, is going to be a very large number. Let's say that it is the log of 10,000. And let's say, hypothetically, that there is only one potassium ion outside of the cell. Because this is a larger number, this will be a negative number. So what I'm saying is, when you're working with logs, the denominator, if the denominator is larger than the numerator, then we will have a negative number, because this is a very, very small number. This is like 10 to the, let's say, 0.00001, right? And 10 to the 0.001 is some negative number. So what I'm saying is, because there is more potassium inside the cell than there is outside of the cell, this is a negative number. If the bottom of the log is larger than the top of the log, this is a negative number. So hypothetically, let's say that this is equal to negative 84. I don't know if it is. I don't really care. I just know that it has to be a very large negative number. So within the eukaryotic cells, typically we have a resting potential of about negative 20 millivolts all the way to negative 200 millivolts. And that's just resting. Nothing is happening, and we don't have an action potential. But the reason why this is a negative number is because there is more potassium ions inside the cell than there is outside of the cell. So now, when sodium enters the cell, it has a positive charge, and this potassium also has a positive charge. When the sodium enters the cell, too much sodium enters, and the potassium is repelled by the positive charge, and so it wants to exit the cell. So if we just look at the potassium, just looking at it by itself, we would have, after the sodium enters, we would have more potassiums outside of the cell and a little bit of potassium inside the cell. So this would be a positive number. And that is why whenever an action potential occurs, it shoots up. It goes to like 30, well, it goes to about 30 millivolts in the positive direction. Because this number is bigger on the top, it has to be a positive number. When sodium enters the cell membrane, it generates an action potential. And the way it does that is it repels the potassium, and the potassium wants to get out of the cell. But you're probably wondering, well, how do we reset this? How do we go back into the resting potential? Because clearly, if I had an action potential that is always being generated, my muscles would be going crazy. Well, yeah, that's true. And the way that it resets itself is that it uses the sodium-potassium pump. So the sodium-potassium pump resets. It resets the cell, the membrane potential. So it resets the membrane potential, and it does this by forcefully shooting out sodium into the extracellular space, and it actively pushes potassium back into the extracellular space. And so whenever the sodium potassium pump takes potassium from the outside and it pushes it into the cell, we get a really, really large amount of potassium inside the cell. And whenever the potassium inside the cell increases, this becomes a negative number. And so we go from a positive number into a negative number. And so what I'm saying is that this resets the membrane potential. It does not create a membrane potential. Actually, it resets it. So it goes back into equilibrium. So we go from a positive number to a negative number. And that is being done by the sodium potassium pump. So on an exam, a answer choice will say, oh, the action potential is created by the sodium potassium pump. And you'll say, no, it's not. It is reset by the sodium potassium pump, but it is not created by the sodium potassium pump. Now, within the cell membrane, you have these potassium leak channels. So within the resting, whenever we are resting and we're not generating action potentials, these channels are always open. And so we can say that potassium leak channels are always open. And this allows us to have a constant supply of potassium. So this is a constant supply of potassium. So now let's kind of zoom in and see how this process takes place. Within an action potential, we have about two phases if you're looking at it from kind of like a zoomed out perspective. So here we have a closed section where there is no current, right? So this is going to be in picoamps, which is very, very small current. So whenever we have an action potential, we have a depolarization. So this is a depolarization. And what a depolarization means is that we go into a positive number. So we go into a positive number right there. Now, whenever we need to go from the open to closed, we are going to repolarize. This is repolarization. And repolarization means that we go from a positive number into a negative number. And that is going to be done by the influx, by the intake of potassium. So sodium leaves the cell, and then potassium enters the cell. And that is going to cause a repolarization. And then we have some time, it would be about 10 seconds over here, where it's not generating an action potential. So this is the closed section, right? So we are not able to generate an action potential. Now notice that this kind of has it has a specific value, right? So within action potential, let's say that we have to hit -50 millivolts to generate an open section. If we hit -49, -48, whatever, we're not going to generate an action potential in this example. And so we have to hit -50. That is the golden number. That is the threshold. And so because we have to hit a threshold, this is called an all or nothing event. So action potentials are all or nothing, meaning that we have to reach the threshold. Okay, so -50 in this example would be the threshold. If I hit -49, -48, that's not really going to do it, right? And you know what? I probably forgot my negative numbers. So -49 is actually greater than -50, so it actually would do the action potential. So what I'm saying is, if you hit -51, -52, something that is lower than this value, then it's not going to work. But if we hit -30 millivolts, then obviously we're going to generate an action potential. Okay, it just has to be greater than this number to generate an action potential. So here is the action potential, and right here we have a -60 millivolts, and the threshold for this system in this example would have to be -40 millivolts. So for instance, -40 millivolts, that's the minimum that we have to hit to get an action potential. So if I hit, let's say -50, I'm not going to have an action potential. If I have -45 millivolts, no action potential. But when I hit -40 millivolts, then I will have an action potential. So this right here is the threshold value. Okay, so this is the threshold potential. Now the resting membrane potential is actually about, let's say, -60. So without any input, naturally this cell membrane would be -60 millivolts, and that's the resting potential. But when I hit the threshold, we have a curve, and that curve itself is the action potential. So this right here, when we are increasing, when we're going up, that is the depolarization, polar. And when we go down, that is the repolarization. Now, to create action potentials, we will be abusing voltage-gated sodium channels. So what is a voltage-gated sodium channel? So we're going to call this voltage-gated sodium channel. So what is that? Essentially, without any voltage or without any electrical input, this gate is closed. But whenever we have some sodiums entering the cell, they generate a little bit of electricity. So they become a little bit more positive, more positive, more positive, until it is so positive that it opens. And whenever it opens, we can actually have more sodiums go into the cell. Okay? So before anything happens, we have little baby amounts of sodium entering the cell. But eventually, the cell becomes so positive that it generates an electrical input. And whenever the electrical input is activated, the transporter is going to be opened, or I guess the channel is going to be opened. Now, eventually, the action potential has to end, and whenever it finishes, the gate closes. Now, whenever the gate closes, we cannot immediately create another action potential. There is something called a refractory period, and the refractory period occurs because this inactivating particle, so this is called, this little tail right there is called an inactivation particle, activation particle. And this inactivation particle prevents this gate from opening again. So you can have another impulse, right? We can get bit by a dog, and really, whenever you get hurt, they send action potentials to the surrounding area, and that generates pain. But whenever the action potential kind of runs out, you cannot have another impulse, right? So my muscles cannot contract for another split second, and they cannot contract because of this inactivation particle. So you cannot have action potentials going like this. Really, it's like this, because this is the refractory period. Okay. And now here we have the membrane potential. So at first we have a closed sodium gate channel. So this is a voltage gated sodium channel, and it is closed. But whenever I hit the threshold, the sodium gates open, and whenever they open, more sodium can enter. And whenever more sodium enters, we have a bunch of sodiums. And when we have too many sodiums, they're gonna take out these little potassiums, they're gonna repel it because they have positive charges, and these sodiums make the potassium leave the cell. And whenever the potassium leaves the cell potential increases, right? So the cell potential increases, and this is called a depolarization. So this is a polarization, right? Now, eventually this has to end, and whenever this ends, the sodium channels become inactivated. So no longer can the sodiums enter the cell. So when sodium tries to, it's going to get blocked. So I cannot enter the cell. But you know what can enter the cell? Potassium, because we're using the potassium pump channels, the sodium potassium pumps, to generate more potassium into the cell. Now, what this picture doesn't show is hyperpolarization. So of course we have this guy right here. So at first we have the depolarization, and this is called a repolarization. Where potassium enters the cell again, but actually the potassium channels, they close slowly. So yeah, we already hit the threshold value. I'm good. I don't need to have more potassiums. But the potassium gate closes very, very slowly. It closes so slowly that sometimes too many potassiums enters the cell. And whenever too many potassiums enter the cell, we have more negative charges. So this is a really negative charge. And eventually, these potassium leak channels will allow us to equilibrate. So you remember the potassium leak channels before, where they're always open? Well, they allow the potassiums to go back into equilibrium. And so eventually, we go back into a normal position. And so this section right there is called hyperpolarization. So this is hyperpolarization, polarization. And I write really, really badly. So this is hyperpolarization. So we have depolarization, repolarization, and hyperpolarization. So depolarization. So this is a positive MV, this is going into a negative MV, and this is a really negative MV right there. And using this picture, we can see that whenever action potentials are occurring, they only go in one direction. So we can say that action potentials are unidirectional. Unidirectional. You might see some textbooks that say propagate, and propagate means to move. So AP, or action potentials, are unidirectional, meaning that they only propagate in one direction. And they only propagate in one direction and not both, so this is incorrect. It would only go to the right side, right? It would only go to the right side because look, we have the inactivated particles. So the inactivation particles, you can't really see it here, but it's blocking the channel. So it prevents the channel from opening. And whenever the channel is prevented from opening, sodium cannot enter the cell. And if we did not have an inactivation particle, then sodium could go that way. The action potential could go to the left, and it can also go to the right. And therefore your muscles would keep contracting, and that you would have a muscle spasm, and that could be kind of lethal within different parts of your body. So thank goodness that we have inactivation particles. So that allows us to move in one direction. So over here we have the action potential being created. So we have the depolarization. It goes into one direction, and eventually these channels will open because sodiums accumulate. They accumulate on this section right here. So the more sodiums we have in this section, they will get near these channels, and the positive charge on the sodium will induce, it will create the channel to be opened, right? So that's why we can actually propagate. So when sodium accumulates on the sides of the cell membrane, it gets near a channel, and the positive charge on the sodium will influence the channel into opening, and that's how we can generate action potentials. So what can we do with action potentials? Well, action potentials can generate signals using neurotransmitters, okay? And so we can use an electrical, we can go from electrical signals into chemical signals, by using action potentials. So the action potential goes from electrical, and it manipulates the synapse into releasing neurotransmitters, and those neurotransmitters can release information. So we go from an electrical signal into a chemical signal, and this is pretty cool. So over here we have these vesicles, and these vesicles, as you can see right here, they contain the neurotransmitters. So this is a postsynaptic membrane, and this is a presynaptic membrane. So this is pre, and this is post. So the action potential is going to generate, it's going to zip along, it will release the neurotransmitters like serotonin, dopamine, whatever, and these little neurotransmitters are going to accumulate between these synapses. And whenever they accumulate between the synapses, they can communicate with the postsynaptic membrane, and they can generate whatever they need to generate. So for instance, if this were acetylcholine, the acetylcholine would go into the muscle cell, which is the postsynaptic membrane, and it would generate muscle contraction. So again, that action potential converts itself into a chemical signal, and in this example, it could generate a muscle contraction. And that brings us to the end of our lecture video. So hopefully now you understand the differences between the glial cells, what the blood-brain barrier does, how the cerebrospinal fluid is created and why it's created in the first place, the anatomy of a neuron, and the differences between the naming of the neurons and the central nervous system and the peripheral nervous system. So remember that I love you very much, and that you are awesome and really, really cool. And nothing is too hard to learn. You just got to put the time and effort into it. Anyways, take care. I love you. And goodbye.